All right, guys, today we're going to try to review a little bit with cell signaling and maybe hit the nervous system a little bit, maybe even the endocrine system, depending on time. The way cells communicate, they do it in a lot of ways. One way is local, and that means when a cell is just talking to a cell right beside itself. And they can do that usually through what are called communicating junctions, the junctions that connect the two cells together. And we talked about two of these. Plasmodesmata are found where? In plants. It allows this plant cell to talk to this plant cell. And essentially it's a little tunnel through what? The cell wall. So they can communicate. They can transport hormones, they can transport materials from cell to cell. What is a similar kind of thing called in animal cells? Gap junctions, or specifically in the heart, it's called an intercalated disc. It's really important in the heart that those cells communicate because they have to what? Beat together. They have to beat in rhythm. That's really important stuff. There's also, back to the immune system stuff we talked about, a lot of cells communicate with each other through proteins on their surface. We talked about class 1 and class 2 MHCs. If you remember, every cell in your body has a class 1 MHC complex out on its cell surface. And the purpose of that is to what? Exactly. Tell other cells, hey, I'm sick, or hey, I'm good. Because what do they put out on that MHC1? They're proteins from inside, intercellular proteins, not intracellular, intracellular proteins. But if they have a viral infection, a lot of those proteins are probably going to be virus proteins, right? And if they do display a virus protein out there on the MHC class 1, what comes around and kills them? Now, this is which kind of white blood cell? Nope.
Which kind?
Killer T cells or cytotoxic T cells. That's called cell-mediated immunity. When your cells kill your own cells that are sick. Cell-mediated immunity. The class 2 MHCs, those are found mostly in phagocytic white blood cells. When the cell takes in a germ, breaks it up, and then displays its antigen, and it's basically sending out signals that, hey, this thing is in us. I found it. I killed it. But there's more of them. You need to go kill them too. And who is that cell usually trying to tell that to? What's the white blood cell that really gets the whole immune system going? The what? The helper T cells. It's basically saying, okay, helper T cell, I found this. You need to be the Paul Revere, and you need to go get the army ready. And you need to tell everybody the viruses are coming. And go out there and get them killed. Another example of local signaling is synaptic signaling. A synapse is a what? A gap in between nerves, but even though it's a gap, it's really a what? It's the way they connect, right? It's a axon to dendrite. It's a connection. And the way that works is that the nerve on the presynaptic side releases what? Yeah, they release neurotransmitters. And those neurotransmitters cross the gap, and they bind to receptors on the postsynaptic neuron, and that starts a what in that postsynaptic neuron? An action potential, right? An electrical signal in that next nerve. So those are all local. Those are cells that are right beside each other, talking back and forth. And there we can see gap junction in the animal cells. We can see plasmodesmata in plants. And this is probably like some cell surface proteins talking to each other, like the MHCs. This might be like a helper T cell and a macrophage or something. They're communicating with each other. I'm not going to deal with that just yet. There's some long distance ways to communicate too. Hormones. Now, for about the gazillionth time, we know hormones are made by endocrine glands. And they travel how?
Blood.
Through the blood. And they only affect cells with their proper way. And we call those cells with a proper receptor what? Target cells. There's also pheromones, which we are usually involved in what?
Pheromone attraction.
Yeah, it's a mate attraction. And they can travel how?
Air.
Through the air, and they can travel for long, long distances. We talked a little bit the last couple of days about plants having hormones. And the plant hormones can travel through the phloem. They can travel from cell to cell through the plasmodesmata. Or which one did we talk about that was a gas? Yeah, ethylene gas. And that hormone, ethylene gas, makes the fruit ripen. And if you put a fruit in a bag with other fruits, what happens? They ripen really fast because what kind of loop gets started? A positive feedback loop where the release of ethylene gas by one apple causes the release by another, which causes the release by another, and another, and another, and another. And it just keeps getting riper faster and faster and faster and faster and faster. Okay. I think the bulb's about to die. We'll have to let it cool off and we'll try again in a second. Not a good time for the bulb to be dying on me. Anyway, let's go a little further here. Let's talk about the 3 steps of cell signaling. What's step 1 in cell signaling? Reception. In reception, the ligand, the hormone, has to bind to a receptor. And most signals are made out of what? Proteins, or maybe amino acids. Proteins or amino acids. So the receptors for those things have to be where? Outside, extracellular. But there's also a few things that have receptors on the inside, and those typically are made from what? Steroids, which are kind of lipid. Which one causes things to happen faster and bigger? The lipid ones, right? The ones that can go inside, because they can go straight to where? Straight to the receptor, and then the receptor and the hormone together can go straight to where? The nucleus, and they can turn on specific genes. It causes really fast, big kinds of reactions to happen. Not so much for the protein-based hormones. But we've got step 1, where the hormone binds to the receptor, and when that happens, what physically happens to the receptor? Changes shape, right? And we call that change in shape a what? Conformational change. So let's look right here. So let's say the hormone comes in and binds to that receptor there. This thing actually probably goes all the way through the cell membrane, and it changes shape all the way through. Here, it triggers something else to happen. And usually it turns on some protein. Looks like that might actually work. Probably going to turn off again, but we'll try it. But it triggers step 2, which is transduction. Remember, the key thing in transduction is that the signal gets, first of all, translated. The cell makes sense of what it's supposed to be doing. And it also gets amplified. One hormone can cause, at the end of the pathway, lots of things to get made, right? It's not a one thing. One hormone can cause bunches of stuff to happen. That's amplification. You got to remember, though, that this transduction step is a lot of steps. It's a series of steps. Let's look at a little bit about transduction. Got that. Skip some of this for a second. All right, but again, like it says here, transduction is a multi-step process. A benefit of it is that a small number of ligands, a small number of cell signals, can activate a large number of molecules at the end of the pathway. That's the amplification stuff. But that whole system of steps is called the signal transduction pathway. And it's sort of like a chain of dominoes. One protein turns on another, which turns on another, which turns on another, which turns on another. And it looks something like this. We'll start here. This is a good example of how transduction works. And this is involving adrenaline, also known as what? Epinephrine. We know that's the fight or flight response, right? So adrenaline comes out. It affects a lot of places in your body, correct? But let's think of adrenaline in the way it affects cells in your liver. What do cells in your liver store that you might need during fight or flight? Glycogen, which is stored like sugar, right? You need a lot of energy if you're going through fight or flight. So epinephrine travels through the blood, bumps into this liver cell receptor, binds there. That's step one, reception. That liver cell receptor goes through a conformational change. And when it does, it ends up turning on just inside the cell membrane this thing called G protein. That'll move across the cell membrane and eventually turn on this guy right here, adenyl cyclase, an enzyme. And the thing you need to know about adenyl cyclase is that it takes ATP and makes cyclic AMP, which is a what? A secondary messenger. Its job is to take the message from the cell membrane to the rest of the cell. But does this enzyme just make one of those? No, it makes bunches of them. And that's what part of this process. That's the amplification part. Because each one of those can relay that message. What they end up doing is turning on a set of protein kinases. What do we know kinases do? They phosphorylate things, which means they transfer what? They transfer phosphate groups from ATP to something, and therefore also transfer energy. So they're turning things on, right? They're activating switches. And eventually, this is going to turn on an enzyme that does what? We're in the liver. It's going to break down glycogen into glucose.
Really excited.
Yeah, maybe. I think we did watch one with him. The pink shirt and the yellow tie. Yeah, I think so. I forgot about that.
I didn't remember if this is.
Yeah, we did. Now here's a kind of an overview of what went on here. The epinephrine bound to the receptor. One epinephrine molecule, one adrenaline. That went through the whole step of transduction, where that one molecule of adrenaline ended up turning on about 100 G proteins. It activated about 100 of them. Those then went out and made about one adenyl cyclase apiece, or activated one apiece. So we're still about at 100. Those adenyl cyclases, it looked like they made about 100 molecules of CAMP apiece. So we're at 10 to the fourth, which is 10,000, right? So from this one molecule of adrenaline, we made 10,000 molecules of CAMP. Those went out and turned on these protein kinases, where at the end of the day, we ended up, I think that says 10 to the 8th. Yeah, that says 10 to the 8th molecules of glucose get released. 10 to the 8th is how much? Yeah, it's 100 million. So there's one molecule that we started with of epinephrine up here ended up causing 100 million molecules of glucose to get released into your blood. That's pretty serious way. Amplification. And that happened through this whole signal transduction pathway. And then the final step of the signal transduction pathway, or the final step of cell signaling, is to get some kind of response. And in this case, the response was to break glycogen into glucose. Does that make sense? So here's a slide that sort of summarizes it. Hormone, or whatever kind of signal ligand, binds the receptor. You get signal transduction, usually involving secondary messengers and kinases, until eventually those kinases end up turning on some enzyme that causes your response. So again, remember that kinases, protein kinases, transfer phosphate from ATP to a protein. It usually activates the protein, maybe helps to regulate it. You've also got these things called protein phosphatases. What do? They take the phosphate off, and therefore take away what? Energy, and therefore what? Turn off stuff. Turn off processes. Inactivate them. All right, any questions on that stuff so far? It's probably about the 100th time we've looked at that stuff. All right, let's talk a little bit about hormones, a little bit more detail about hormones. With the endocrine system, there's maybe 4 or 5 hormones that I really want you to know a lot about. There's way more hormones than that. I can't guess which ones they're going to ask about, but I know in the past the kinds of things they've asked about in terms of the endocrine system. So I want to use that. And one thing that we need to talk about is the whole controlling blood sugar. What gland makes hormones that control blood sugar? The pancreas. So let's say that here's homeostasis, and let's say that you ate something really, really sugary. So your blood sugar levels start to spike up. And the pancreas says, okay, I can take care of that. And the signal it sends out when your blood sugar is too high is what signal? Insulin. And that insulin travels through your blood, and it specifically targets cells in the liver, cells in the fat, and also muscle cells. And the message to all of those cells is to do what with sugar? Take it out of the blood and store it. The liver and the muscles take it out and store it as what? Glycogen. The fat cells take it out and do some fancy chemistry on it and convert it into fat. But regardless, the message insulin is sending, take insulin out of the blood, store it, so that blood sugar goes back down to our set point. Now, on the other hand, if blood sugar is too low, maybe it's been a while since you ate, your blood sugar levels drop. Pancreas says, I can take care of that. So it releases glucagon. Don't confuse that with glycogen. Glucagon is a hormone. Glycogen is what kind of sugar? What kind of saccharide? A polysaccharide. Glucagon goes out to cells in the liver, maybe in the muscle, maybe in the fat. The message that it's sending to those things is to do what to that glycogen you've been storing up? Break it down. Release it into the blood. Raise my blood sugar levels. Those two hormones, glucagon and insulin, are opposites. And what do we call hormones that regulate something because they do it in an opposite direction? Antagonistic hormones. And there are lots of those in your body. Antagonistic hormones. All about maintaining homeostasis. A couple of other hormones that I want you to be familiar with. Your throat. What's the gland in your throat? Thyroid. And it makes 2 main hormones. Anybody tell me what those 2 hormones are? Yeah, T3 and T4. And T3 and T4, those 2 hormones help to regulate what? Metabolic rate, the rate of cell respiration. What are the 3 and 4? What are they referring to? Again? What element does the thyroid need that's only needed by the thyroid? Your body doesn't need it anywhere else. Magnesium. No. Iodine. Right? And that's T3 has, guess what? 3 iodine atoms. T4 has 4 iodine atoms. But regardless, sometimes these 2 together are called what? As a group. Sometimes they're called thyroid hormone. Sometimes they're called thyroxine. What cells in your body have receptors for thyroxine or T3 and T4? Pretty much all of them, because they all have to be able to do it. Cell respiration. And what's the message that T3 and T4 are sending? Speed it up, speed it up, speed it up. When might you need to do that? When might you need to speed up respiration? Say again? Yeah. Say you're exercising a lot, you're at the gym, you're working out, you need more ATP, or maybe you're outside and you start to get wet, start to get cold. Your body needs to speed up metabolism because metabolism is really what? What are you doing to sugar during metabolism? You're burning it, and what is that going to release for you? Some metabolic heat. That's the way to warm yourself back up. Speed up the rate you're burning things at. So whenever you hear thyroid, think speed up metabolism. Your pituitary gland makes a bunch of hormones. By the way, where is your pituitary? In your brain, just below what really important part of the brain? Well, it is below the cerebrum, but the hypothalamus is what I'm thinking about. And remember, the hypothalamus is sort of the connection between what two important systems in your body? The endocrine and the nerves. It connects them together. It's part of both. So that's huge. The pituitary, two hormones I want you to think about when you think about the pituitary. First of all, HGH. What is HGH? Human growth hormone. When do you release a bunch of that? At puberty, around puberty. And what cells have receptors for it? Pretty much all of them. Now I just gave you two examples of hormones that pretty much all cells have receptors for. Is that usual? No, usually not. But all cells have to be able to grow, because what if only your bone cells had human growth hormone receptors? Your bones would grow, but your body would be a little out of proportion if that happened, right? It would be a little weird. If your bones got longer and the rest of you didn't grow with the bones. So you have to grow proportionally. Another hormone that I want to make sure you know something about is this one. ADH. And that stands for anti-diuretic hormone. What's a diuretic? Yeah, makes you go to the bathroom. Well, this is an anti-diuretic. So it keeps you from urinating out a bunch of water. First of all, what would the target of that hormone be? The kidneys. And when would you want to have that hormone? You're dehydrated. And if you're dehydrating, do you want to be peeing out a bunch of water? No. So you need to conserve water. And this makes you reabsorb that water from your kidneys back into what? Where do you want to keep the water? In the bloodstream. So absorb it from the kidneys, put it back in the blood, keep it. So your urine's going to come out how? Really dark, really concentrated, as little water as possible. When would you make that? Other than, well, when would you make it? We said dehydrated, but this could be in response to like exercise, sweating. It could be in response to having something like diarrhea, vomiting. Anytime your body is losing a lot of fluids, your pituitary is going to make this ADH. And it's going to help you to keep in as much water as possible and try to keep you from dehydrating. Does that make sense? All right, let's see if there's anything else here we need to look at. Let's look at that one. This is something we talked about in class a couple days ago. What's that little plant stem doing? It's growing toward the light. What do we call that? Phototropism. A tropism is just a movement of light in response to, not a movement of light, a movement of a plant in response to what? The environment. Could be to light. What else can plants move in response to? Gravity. Touch. Yeah, that would be touch. Yep. In this case, I told you there were two plant hormones I wanted you to know something about. One of them was ethylene gas, we talked about a minute ago. The second one was a group of hormones called auxins. What's the message those auxins send to a plant? Grow. Grow, grow, grow. But what stimulates a plant to make a lot of oxygen? Shade. So in this case, when this plant is growing that way, growing over toward the light, where are most of the auxins at? Over here. And you can see that this side over here is growing faster than this side right here is. And that causes it to bend toward the light. That's the reason it bends. The shady side is growing faster than the side on the light. And that causes it to bend to the light. Makes a lot of sense. All right, let's take a minute and look a little bit at nervous stuff. And we'll call it a day on this. All right, so the basic nerve cells, a neuron. And they've got these little tree-like extensions coming out, these guys. And those are called dendrites. What are dendrites usually connected to? Either the axon of another nerve, or maybe some kind of receptor, like a touch receptor, pressure receptor. But what do dendrites do? Incoming information, right? They receive, they're like the receivers. Incoming information. This part here in the middle, where you see the nucleus, that's the cell body, or sometimes it's called what? Soma. And then usually most neurons have one axon. This whole thing. And it carries what? Outgoing info. And it's usually coated with this lipid-like substance called what? Myelin, which is a kind of insulation. But remember that myelin sheath has these little gaps in it. And my marker's off a little bit, but you can see what I'm going for here. Going for those little gaps. And those little gaps are called nodes. The action potential, the nerve impulse, will actually jump from node to node to node to node to node, right? And why would it want to do that? It makes it go fast. Instead of having to go through the whole thing, it jumps. And that makes the process of conduction a whole lot faster. So here's an axon, the end of an axon, and this would be what right here? That'd be the dendrite. So this is a dendrite on one nerve and an axon on the other. This would be the presynaptic neuron and the postsynaptic neuron. And then this is the synapse. And here we're seeing that a little bit closer up. So an action potential, and remember that's just an electrical signal, right? Electricity, action potential, is moving down the nerve. It gets close to the end of the axon. That causes gates for what ion to open up here near the end of the axon? Calcium. Ca+2. And those calcium ions rush into the nerve and essentially push out what? What's in those little vesicles? The neurotransmitters. They cause those to get pushed out. And the neurotransmitters flood into the synapse and they diffuse across it and they bind to what over here? Yeah, receptors. And those receptors are called, in a nerve, are called ligand-gated, what's going, ion channels. And essentially what happens when those neurotransmitters bind, what do they cause to happen to those ion channels? They cause them to open. And when they open, what floods into this postsynaptic neuron? Sodium ions. That's the beginning of a what? Of an action potential, the electrical signal. So it's not that a spark of electricity jumps from here over to here. It doesn't happen. The neurotransmitters cause these gates to open, and those cause an influx of sodium. Sodium ion has what charge? +1. That's a moving electrical particle, a moving charged particle. And by definition, what is a moving charged particle? That's electricity. That's an action potential. So let's talk about a little bit about action potentials and we'll be out of here. Mr. Anderson, in the video we watched, used a good analogy to help you remember how a nerve is set up. What was his analogy? Salty banana. That salt, let's say salt on a banana might be a better way to think about it. Because the salt refers to what? Sodium. When a neuron is set up to work, where's the sodium at mostly? Outside, around the nerve. We have lots of sodium ions. And then the banana part, what's there a lot of in bananas? Potassium ions. So those are inside the neuron. For a nerve to work, it needs to be set up this way. But what's it called when it has this setup? So it has one thing in one place and one thing in another. Polarized. The neuron is polarized. It's ready to work. Sodium and potassium are separated from each other. Now, when a neurotransmitter binds to this neuron, what did we say that causes to open up? Sodium channels. So sodium begins to rush in. Why is it going to rush in? There's a gradient, right? And things always want to move what way? High to low. And if it's going through a channel, what kind of cell process is that? What kind of cell movement? It's passive, but what kind of passive? Facilitated diffusion, right? High to low through a channel. And that sodium rushes in. Sodium, sodium, sodium. So more and more positives. Remember that this neuron starts out with a charge of about -70, what's an MV? Millivolt. But somewhere in here a stimulus happens and sodium starts to rush in. So what happens to that voltage? It's positives, right? You're putting positives, so the volt starts to go up and up and up and up and up and up. Now, the cool thing about this is what, let's say this is one node, and then we got a little gap in our myelin, and then we got another node over here somewhere. When the voltage over here gets to a certain level, what does it cause to happen at that next node? It causes the sodium channels over there to open because they're gated by what? Voltage gated, and they open up based on charge. So this thing, that's how it jumps from node to node. When the charge here gets to a certain level, it causes the sodium gates over here to open. And eventually this will cause the sodium gates in the other place to open. Anyway, sodium continues to rush in until the charge gets up to about +40. And when it does, what happens to the sodium channels? They close. And what's triggered to open? Potassium channels. And which way is potassium going to move if it can move? It's just going to go out. So as the potassium starts to rush out, what starts to happen to the voltage inside that neuron? It decreases. It goes back down. And eventually it gets back down to normal. But when it gets back to normal, is it ready to go? No. Because now what's the setup? Yeah, now you got potassium mostly on the outside and sodium mostly on the inside. Is that the way a nerve needs to be to work? No, not polarized the way it needs to be. So what protein will fix that? Sodium potassium pump. It always takes sodium and actively transports it out, and potassium it actively transports in, so that we get back to that polarization event. And that's what's going on in this little dip in here. Go ahead, Em. They're specific. The sodium gates only let sodium in. Potassium gates only let potassium out. It's like a selective tunnel. It's not just a hole. It's like grabbing them and letting them go through kind of thing. Hey.
it's not good. You got it.
That's not
But
good.
he's been trying to make it.
Okay, so I'm hoping it feels good. Anyway, that's our action potential. And essentially what we're looking at is what? What is that? I mean, it's an action potential, but what does that mean? It's an electrical signal. If you looked at an EKG, what would you see in terms of that? You just see it over and over again, right? That's how your heart works. It's sending this regular occurrence, this regular sequence of electrical signals over and over and over and over and over again. All right. Make sense? Let's see if there's anything else here we need to look at. That sort of summarizes what we just said. In an action potential, neurotransmitters from the presynaptic neuron bind to a ligand-gated ion channel receptor on the postsynaptic neuron. This causes an influx of sodium ions into that neuron and starts the action potential. The voltage-gated sodium channels continue to open further and further up the neuron and from node to node. When the voltage reaches, depending on the neuron, either plus 55 or plus 40, the sodium channels close and the potassium channels open. Potassiums rush out, and eventually the sodium-potassium pump gets things back to normal. That's an action potential. And then the very last thing we will look at, reflexes. A reflex is important because it skips what? It skips the brain. You have reflexes because sometimes you need things to happen how? Fast. Your brain's a computer. It has to process things, right? Stuff comes to your brain, it has to process things. Some of us process things a little faster than others, right? It takes some time. If you're about to fall flat on your face because your foot got stuck on something, you don't have time to process that, right? So your brain has certain things hardwired, like that reflex that when your knee tendon is stretched or it has pressure on it, what's the reflex? Kick. That keeps you from falling flat on your face. Or if you touch something really, really hot, jerk it back, okay? So a reflex involves a sense neuron carrying information directly to the spinal cord. That neuron is connected through what's called an interneuron straight back to a motor neuron. So it's like hardwired. If this happens, then do this. It's like an if-then statement. If this, then that. And it's quick. Brain's not involved. Keeps you from getting hurt. I'm going to stop there.